Okay, it's working. Microphone, okay. So I want to, we're still waiting for people to join in. And I want to first describe a little bit about the arrangement of our student presentation and the final exam. So today is the let's see, arrangement. So we have, so today, so we will do manifold next week. And then network the week after. So then the lecture is done. And then this week, you will come here to the student presentation. So this will be either a project or a proposal of presentation of your design study or your finished study, whatever. So we need a significance. So the topic you select had to be biologically or computationally significant, not just a present paper. Other people help you identify the problem and significant. And some of innovation, that's an aspect that we want to look at, and also the effectiveness of the delivery. Okay. Easily, you explain the complex concept to students who has not been able to, not time to think about the problem at present. So that's the student presentation. The final exam will be the week after. Then this can be done remotely, or if you decide to do it at home. So this is just a multi-choice questions. That's our arrangement. Okay, today we have a couple of distinct problems, topics. I will mention batch effects. So that's where you have two different data sets. And then we will discuss, okay, CCA is one of the batch correction method. We will talk about pseudotime trajectory analysis. To see the applications and the meaning of this analysis, and also commonly used packages and underlying methodology. Then I will talk about highly variable genes. This topic, the highly variable genes, I believe is underappreciated because people use it as a filtering to see, okay, I want to find marker genes and that express differently across populations. But when you consider the highly variable gene within a homogeneous population of cells, you don't use it to differentiate cell types, but you characterize cell states okay, because they are same population of cells, same cell type. And so still you have this highly variable genes. Sometimes you call gene expression noise. Now we'll show you this gene expression noise actually have information about the function of this gene inside this type of cells. So that's the topic we are going to cover. Yeah, so. So what's the batch effects? So we have control and the treatment and the two different sets of cells, and you measure the single cell expression analysis using X genomics, for example. And you get those cells once you plot them in Kisney plot, and you see, okay, they most likely they will be separated into two different large clusters, okay? In this case, if you believe there's a good reason that they shouldn't be like that, then you can do the batch effects correction. But I have to warn you before we move on. More and more people, including a lot of senior members in this field, believes batch effects correction is unnecessary, okay? Unless you have strong reason, you believe you have batch effects because your sample one may be sequenced or sampled in one lab or in different time, and your treatment of the same group was carried out in another lab, different locations, and use slightly different conditions, which should be considered as experiment bias. Then when you see this separation of control and the treatment group, you believe they shouldn't be like that. Now, the difference shouldn't be so substantial. Then there's a justification. You want to do the batch effects correction. And the result of the correction is you align your data set and merge them. Okay, you correct those, because if you now use unaligned data set, when you do the differential expression analysis, a majority of the cell genes may be differentially expressed, thousands of them, but after you align, you will correct those inconsistencies, and then when you detect anything difference between two groups in the aligned data sets, and those could be the real signal that differ, make two cell lines different before and after treatments. Okay, so that's a justification. Batch effects correction is not a new problem, not only limited to single cell data, but basically for any experiment in biology, you have to consider whether what you see is from a real difference of the biological system or from the batch effects introduced through the process of experiment procedures. If you have a strong reason, again, you believe these batch effects give you the confounding effects you want to control for that, then you can use those methods. So before single cell data error and the comeback is one of the mostly commonly used method or packages people use to correct the batch effects between samples get from a traditional RNA-seq analysis. People use this for microarray data as well. So you have compact. Then when single cell become available, people develop the proposed MNN, mutual near neighbor, and the CCA and other methods to be considered as the improved method which apply to single cell data to take into account the nonlinearity between data points. Okay, so how can we do that? There's a study, and people start to compare this original methodologies developed for the traditional RNA-seq data and see whether I can just apply those to single-cell data. At some level, it's okay. It's still correct a lot of stuff, and come back, still be able to use it in this case. When when situation become more complex, because you have multiple cell types, complex relationship between cell types. So that's why there's a new methodology coming in, and to deal with this level of complexity. So side by side, there are two papers published in Nature Biotechnology in 2018. The first methodology is called mutual nearest neighbor batch correction. So this method is designed in this way. So first, you want to look at, so you embed your cells, of course, just like you did in UMAP, your Disney, and you embed them for different batches. So in this illustration, you have batch 1, batch 2, and then you start to look at cross batches, the nearest neighbor, just compute the distance between cells. Okay, so if you start from batch 2, for example, you start to look at those right cell and look at its nearest neighbor. If you found, okay, the nearest neighbor, and then the other way around, you also found the nearest neighbor of the batch 1 data to batch 2 data. So they call the mutual pairs of those nearest neighbor. But it's just in the embedding space, they are closer to each other than between them to the other cells. So you can make a link between them. In this example, in panel B, they found, okay, those cells are nearest neighbors, and those blue cells cross batches with the blue cells, they are nearest neighbors. And once you have those relationships, you can start to merge, okay, merge the cells. Okay, so you just put them together numerically and project them onto the shared space. And in the same time, you will drag other populations to the same space. So looks like there was 3 dimension. Okay, batch 2, batch 1 are separated by the distance at the z-axis. But after you pulling this down and project them onto the same space, the 2-dimensional space. And those yellow ones, because there are no mutual neighbors, there's no other cells to be grouped. They will be as a distinct group as they are. Okay. And later, maybe you have batch 3 and it comes in, and you can redo this to embedding and the reduced dimension to merge cells. And in this case, batch 3 show the mutual nearest neighbors, cells with yellow cells. And then you start to merge batch 3 and to the shared space. So that's a distance-based method based on the KNN. By the way, the K, it's not a KNN. K, so K is the number of nearest neighbor you will consider when you do this, right? And I said side by side with another paper published in the same journal. And these authors use CCA, canonical correlation analysis to do the batch effects correction. Again, to assume before and after treatment, this unaligned data set contains unwanted confounding effects because of the process of this experiment. So if you have this concern, you can use the CCA to project them and to find the largest correlation between two batch effects data. And then once you project them onto the shared space, you found two rotation matrices, one for batch one, another for batch two, and you rotate your data respectively, and then the shared projection will be mostly correlated. And then you use so-called dynamic time roping and to correct for this different scale problem, because you see, okay, the projection of the cell one looks shranked into a narrow space, while this Ceron cells looks like more spread out. And you can use this technology to realign them. Then at the end, you correct your T-SNE, and then to move the cell around to make them embedded in the same space. And they basically align and then ready for the downstream analysis. Yeah, I want to check my meeting. Make sure I can hear you. Okay, good. Okay, let's spend a couple minutes to look at technical details for this CCA. So canonical correlation analysis is a method of correlating linear relationship between two multi-dimensional variables. Okay, make use of two views of the same semantic objects to extract representation of the semantics. Okay, so you have to underline assumption that two data sets are related somehow by this semantics. Okay. Let's see. So you will choose rotation matrices for two different sets. The sets maybe you consider from a same semantic object, and then you have different views of this. Okay. Apply to single cell data is we consider the cell population defined after treatment. It's still the same semantic object, but you just view them from different measurements, and then we have different projection and in your collected data. So, but they should largely overlap. So if you have two views, you will do this CCA and to rotate them. So the correlation between them in the shared common feature space will be maximized. So what is semantic information? So you can have for the same person, you can have a picture of her, and also you have the sound, the voice recording signal from the same object. once you have enough of this, you want to do a kind of alignment. So can you do this semantic space projection so you can make a link between the picture and the soundtrack information? So this is one of these examples. Another example is from this database. So you have pictures, and then you have verb text description of what's in the picture. So that's the view of the same object. One is use pixel, the image, and then another use language character description. So the two different views. If you have a way to project their features in the same space, you may be able to align them. Another example is this Eeg and Functional Mri. So they have different measurements for the same brain neurological activities, and then you want to build a correlation between them. Those are just examples, and also every kind of data set here is high dimension data. Okay, we are dealing with multi-view, high dimension data set problem. How do we do this? Okay, for example, you have data set one. So this is like view one from the same object you sample the data from. And this variable, shouldn't be variable, it's a data set one, okay? So, so for this, as PCA, we can find a rotation matrix for it. And then we can project and rotate those data points to some new angle, right? We can do the same thing for green dots. So that's view two from the data set two. We can do the same thing for that. That's what exactly CCA will do. CCA will find the two rotation matrices, one for data set one, one for data set two. In PCA, we just care about just one data set and look at the rotation afterwards. You have a more spread out of the data. That your projection for PC is the largest compared to any other rotation matrices can get. Well, here is a difference. It's not a concern about the most spread out of any individual data sets. Well, it will focus on your projected data in data set one and two are mostly correlated afterwards. So you have a reduced dimension of data set one, you have a projected data, and you will do a correlation between your projected value with projected value of data set two and make this regression, okay? So if you have established a regression line which can explain the correlation between dimensionality reduced data set from data set one and two, then you have a strong correlation between them, then you are done, okay? So you will find the rotation matrix for data set one and two to make sure after projection their correlation is maximized. Yeah. You don't have to be overlapping to each other. If you have different scales, and one is, for example, one is a kilometer, another the miles, and you do have a separation, but if you projection afterwards, you always point the structure, always point to the same direction, you should have a strong correlation between them. So in order to better understanding this correlation, you will want to know how you compute the correlation coefficient. Correlation coefficient, for example, rho here, is actually almost the same as covariance matrix. Okay, covariance matrix, you have done covariance matrix. And also, if you have two data sets, you can compute the covariance matrix between variables, between those two by this inner product between two matrices. Well, correlation coefficient is just normalized covariance matrix. I mean, covariance between two variables. It's normalized by the variance of dataset one variable and also dataset two variable. Okay, so that's just structure of this. So eventually, you want to maximize this part. And you also, at the same time, minimize those and make this covariance matrix larger. The covariance is between two projected data sets. And then in the same time, you know, make this variance smaller. So this algorithm is simple. You want to have matrices. In this case, will be A and B. Okay, projection matrix. After you use A to project data X, and use B to reduce dimension and project and rotate data Y, and then the correlation between this rotated data and become maximum. And you found these values in A and B. So you want to change the values in the rotation matrices A and B to make the final correlation maximized. Okay. So this final correlation of this can be expressed in this way. Eventually, you get this. Okay. Once you have this formula, you know how to write your, if you want to do this as an optimization problem, this will be just the cost function. So you want to maximize this, which means you want to minimize the negative of this, right? Well, linear algebra found a solution which you don't have to go through optimization. You just use SVD to do the decomposition, and then you learn from there how can you get A and B. So this is a linear algebra solution. That's MATLAB function. This will just get two input matrices. It will return A and B. Okay. A and B will help you to rotate your X and Y. Okay, they have an output. And R if you need a correlation. So R will be correlation after you finish this how good you can get. So this is a link between CCA and the PCA, and another two similar technology is partial least square. It's one of the regression algorithm, and also multivariance linear regression. The reason you can put them together because they all follow the same kind of framework to do this lineage algebra solution. The only difference is the matrix A and B are different. Okay, for CCA, yeah, for CCA, you can consider A and B matrix are those. Well, in PCA, you only consider this covariance matrix, basically. And the B is reduced into an identical matrix. We will see this is similar, unrelated problem like dimension reduction, PCA, CCA aligned data sets, and also PLS, MLR, regression. They all internally linked somehow under the same kind of eigenvalue problem. Okay. Now just come to optimization solutions. Yeah. In this framework, instead of using this correlation as the loss function, looks like this solution and check the difference between this. Just like Alex mentioned, okay, you make the distance smaller in the shared space, also the solution. Right? I hope you can read this right now after those weeks. So this is to use A as a rotation matrix, rotate X data. And then here you use B as a rotation matrix to rotate Y. X and Y are the two input data. And then you want to find the difference between them to make them as smaller as possible. So this is a norm, and this is a squared norm. Where you want to find optimized values in A and B to make the overall as smaller as possible. So that's optimization solution. So what we said so-called conventional CCA. There are some assumptions, and you have more samples, enough samples to give you the estimation of those variables. And also you assume largely you have normal distributed data, the variables. So also this linear relationship. If not, then you have other sideways selections different. For example, you want to transform data, use a kernel function remember kernel. So we have this data is here. You increase the dimensionality by taking some computations through the kernel function to make them into a kernel space, and they become separable. So in this case, you want to use kernel function to pre-process your raw data and make them separable, and then all from a nonlinear relationship become linear, and then you use CCA. So in that case, it's called a kernel CCA. And there are also sparse selection methods in this way. Okay, this slide just illustrates that we can use the kernel function to transform data, okay, from the original, and it becomes even higher dimension data. Okay, the reason for that we want to introduce this kernel transformation is because we want to make them separable or more linear in another space after transformation. Okay, here is just another CCA, which is slightly unrelated. So this is called the canonical correspondence analysis, and the people in ecology community using this technology in their particular domain solutions. So don't be confused. Okay. Nothing, I just mentioned this. There another CCA, which different group of people are talking in the different languages. Okay, as I said, we mentioned this when CCA applied to single cell data, that Nature technology paper. Last step, they use the so-called dynamic time warping technology to finalize alignments. So this technology is to match, basically, to optimize to given time series sequence. These two sequence data should not be the same length. One may be shorter than another. And then you will use this technology to stretch the shorter ones and maybe compress the longer ones. At the end, you will make them match spatially. The example of using this is at the right side. You see, okay, I have two data sets. They all time series related. So this blue curve, spiral curve, and the stretched much longer. So we have from 0 to 1000, where this orange curve and it's much shorter. Okay, the data points on it is less, so you have x, y. So after you finish this time warping procedures, and then you actually align this yellow curve along with this longer blue curve. So you can see blue curve largely unchanged in the shape, and also the density. Well, this orange curve and the data points become more spreaded, and because each of these found its relative aligned points on blue curve. Okay, so you make this densely sampled data and with this sparsely sampled data comparable and matched to each other. So through this procedure. So I just mentioned those methodology, the name of this methodology, and I hope you can get the idea. And once you need that at a certain time or in your research in the future, and you have those kind of data, so I want to align this, what kind of technology I should use if this dynamic time warping technology is come onto your mind, and that's the goal of the lecture. You don't know when you will have those situations. Okay, so this algorithm is based on the distance first. You build a distance matrix between two time series data. So one is A and another B. So all the data points will be the data points in A and B will be put in the row and the column and columns. Then you compare the pairwise distance from any given point in A to B, and then you build this matrix, distance matrix. And then you will use a technique called dynamic programming to search the basically to do alignment. If you have taken bioinformatics sequence alignment course, you may have that experience. You search along this is the matrix and try to move from this lower left corner to the upper right, and then to find the shortest path by cross these values and make the sum of the value as small as possible. So that's the solution. For example, you move from here and then you jump to the diagonal, then you move up 2 steps, move to diagonal, and then move to the right 3 steps. So based on those paths selected, and then the total, the sum of this distance value becomes smallest you can get. Then you just follow this path to map the data. For example, you move 3 steps in blue curve. That means these 3 steps, 3 data points next be mapped to the same location, which is still the second point in red curve. Well, after that, you build this correlation map, data points from one curve to another. That's your solution. So it's a distance based, and it's based on this distance matrix and the dynamic program. There are some studies to compare the performance of different batch correction methods. Then, because we don't have a ground truth, we don't have a golden standard to see, okay, this method is better than that. But through the visualization, looks like there's no clear cut. So I have the best solution, and also we found, okay, combat is also useful and give reasonable solution compared to more modernized technologies. Okay. Okay. Do you have any questions for this battery effects correction? CCA is okay. Okay, so we move to trajectory analysis. So this this idea is from cell biologists, not limited by tracing cell lineage. Not you only use single cell data, but long before the single cell data become available, people started try to understand through the development stage, for example, how individual cell or cell lines, the linkage are linked, and we try to trace back to reconstruct this dynamic cell development differentiation procedures. And there are many applications. You may be able to follow, for example, some mutations in DNA, or using some markers, GFP, to follow if you have a zebrafish, maybe it's larvae or embryo are transparent, and you can trace down some of the cell lineage across the development time. So those are the technologies and before single cell, and the biologists can use to do this so-called cell lineage tracing studies. Single cell data now gave a new chance to look at, because you have sampled from a single tissue tens of thousands of cells. Those cells may be at different development stage in terms of this individual animals. Sometimes like this scar formation or regenerative biology have samples and the damaged tissue and the healing process. You will see those cells in the bone formation. So you will see the cells in different differentiated stage. So even a single sample, you may be able to find the cells in this different stage. You want to make sure you will be able to reconstruct the history of individual cell lineages. Okay. So the idea here related to canonization, and this is a Latington and proposed this canonization idea to describe, to characterize the genetic robustness in the cell line system, not cell line system, but in general, animal development as well. So the idea is you may have different mutations in the population of the species, but eventually, so this variance phenotype will not show up, because the canonization effects will correct and only allow some of the solution. So you apply this principle to cell differentiation. You will say, okay, if you have a stem cell in the early stage of development, this is immature, and you have different ways to go, but eventually you go either this cell line or that cell line, or that there's only a couple of stable solutions. Okay, you can eventually commit to that particular trajectory. So those trajectory, we hope we can use single cell data to reconstruct. Okay, if you're not enough for cell population from one individual, you may be able to collect data from multiple individuals which develop at different time points, but you pull cells in together. So that's the whole idea about the trajectory influence of pseudotime analysis. you want to reconstruct gene dynamics and relate it to the cell fate or cell trajectory development or differentiation. Okay, so because of single cell data, you have normally use dissociated cell population, so we lost the temporal information. You can certainly collect data from different time points. Still, within the same sample, you don't have those temporal information. So that's why we want to use pseudo-time estimation to reconstruct this trajectory, allow us to then, using this statistically inferred ordering of the cell, use them as a pseudo-time series information, and then you then bring into the system a new axis. So now we use pseudo-time as your x-axis to look at the expression of gene among the cell aligned by those orders. And you can understand the role of different genes in this process. For example, gene A downregulated across the development time, will the B as a function of the time have an increased level of expression. Then this A and the B genes are pseudo-time associated genes. Well, other genes maybe not have this pattern or more complex pattern you can look at. Okay, that's the principle of this. We will look at so how people use it as an example, and this for the stem cell to develop into endothelium cell differentiation. I think this is one of the early study using single cell data. Then you can have this reconstruct trajectory and then follow them. You can see, okay, from endothelium branch, which those genes are responsive. Well, in another branch, this mesenchymal branch, and different set of genes get involved. So that's one of the application. Then you can look at whether there are transcription factors, which are the responsive gene, which under regulation of these transcription factors. Once you have enough information, you probably can figure out different parts of the development stage, and also responsive genes. Okay, this picture I just want to further illustrate, and you can use a single cell data to look at the dynamic process. If you think, how can you get this picture, and you certainly have to take multiple pictures for the same banana at a different time and then reconstruct this. We just cannot follow the single individual cells because the technology is disruptive and we lost the live cells. In that case, we take a picture for the population, okay, a lot. Then we align them, and by the similarity, more similar cells put as neighbors one another, and assume that we can reconstruct this smooth movement of the video, although it's not, it's from the reconstructed from the population. So it's very likely if you have a T-SNE plot, you have a group of cells. Well, you want to make them somehow linearize the cells, okay? Assume you follow a pseudotime. Each cell then will be assigned with a value from you can rescale it from 0 to 1, whatever artificial scaling you can use. So this called a pseudotime, is not related to any real biological observation yet. That's just a game of the data analysis. Okay, then this information can be also incorporated with cell types. So now we have a projection which can cross cell types. It will be relatively easy if you use only the same cell type, and then you just make the projection elongated, and then you see, okay, this end the cell similar to each other, that end the cell similar to each other, and in the middle there something, the cell in between, and that's the state change. But in this case, you have multiple cell types. You have to find the right trajectory, and you know from the cell development knowledge and which cell type derived into another and which is the mature states. So you have to work out this if you have multiple solutions to see which trajectory you should follow. Most likely you will get a network between different cell types, right? And in this case, you really need your biology knowledge to get in. So you want to go this way, or you want to follow the trajectory along the network of cell populations, go which way you want to go, okay? And that will have an impact on your downstream analysis, because if you have this pseudo-time arrangement differently, and you get a different picture of this. And for this particular arrangement, the ordering of cells, you get, okay, there are downstream, down regulation of those genes, and up regulation of that genes. But if you select a different trajectory, you may end up with a different set of genes. So in this case, data science has no good solutions. I have no idea whether we should have an early stage stem cell maybe, and what's the late stage next movement. So really, although we have this methodology all available here, your understanding of your cell systems at the end will be the key. It will allow you to select the correct trajectory and to analyze downstream. Okay, let's take 5 minutes break. Okay, let's move on. So as I mentioned, what we need to do is to start from embedded cells, the embedding of cells by t-SNE or other method of TCA. We'll have to allow them to collapse into a certain particular trajectory, which the trajectory may be linked across all the cells and go through all the populations of cells. So those different software solutions packages and use slightly different procedures to do this, but eventually they all do the same thing. And the first step is always to do a dimensionality reduction, so you get the projected cell populations. So you get this for the start from same position here. And then, for example, Monaco, remember this version 1, and use minimum span tree to find the centers of the clusters, and then create this tree structure. Okay, so the minimum span tree means you found a tree which is a link between all the subgroups of cells, but the total length of the tree branch will be the smallest to, but all the cell population will be connected. So the slides also use the minimum span tree construction, but only look at the cluster centers. and then use some kind of a entropy T-scan, and waterfall use same minimum span tree, use the cluster centers, and SCARBI, and use curve fitting. Okay, so mostly you have a curve to fit all the population of cells in the embedded space. And then the next step is to collapsing individual cells, because you have the tree or trajectory, but the cells has not been on the trajectory yet. So you do some projection or collapse those cells, just move a little bit to the nearest trajectory points. That's all. Basically, whatever method you use, you are doing the same thing, and there's no biological justification in there. Just play around with data points, and then you try to figure out the biology later. So this image show you the minimum span tree. You want to have centers of these clusters, then you have to compute the distance between centers. At the end, you make a tree. This tree will link all the nodes. So the age, the total of the age which connect those nodes will be the smallest. Another method, which is slightly different from a minimum span tree, is adopted by this packages Wishbone and Wanderlust. They use nearest neighbor graph, basically the KNN network between cells. So once you have this network, you will capture the local similarity between cells, because you move along this network, you can find those trajectory link with everything, but not a jump from the red cell directly to green cell, because you have the restriction of the nearest neighbor graph. You can only move by small steps to navigate this complex relationship between cell populations. At the end, you have this selected paths along networks, then you collapse cells onto this trajectory. And this is an example of nearest neighbor, and you may be able to click the link if you like to get this demonstration how this nearest neighbor network can be constructed, and also you can change the parameters. If you use k equals 4, you get this. If you use different k equals 3, then you will have this isolated network, because all the 3 neighbors, they are next to each other. But if you have a k equals 4, this becomes linked. So there's not a difference. It's impacted by this, the k values. This we will try in MATLAB, and you can build very easily with, once you have embedding, you can create this nearest neighbor tree. And I also here I use curve fitting, and then you can look at the different genes which have a up or down regulation and along the pseudotime. If you assume this fitted curve as a pseudotime, and you start from one end of the population and then move to another end. There are other algorithms. I don't think I have time to go through them one by one. But overall, the goal is the same. I have to mention Monaco, because this is widely used as one. By the way, I don't like this package of the parties. It has 3 different versions, Monaco 1, 2, 3. And they're almost completely different algorithm underlying. So the author shouldn't use the same name across all the history of development of this. It's get confused because you have to mention you use Monaco version 1. That means you use ICA and the minimum span tree to do this. Well, in Monaco version 2, they use so-called reverse graph embedding. So I would like to say to the author, if you use completely different algorithm, you shouldn't. And then that's a different version. So anyway, it's widely used. And this reverse graph embedding use so-called principal graph, and similarly, like a principal curve that goes through the middle of the data points and to create those branch. Okay, so that's a solution for Monaco 2. Then they come up with a Monaco 3. I don't know the link between this. I have no idea whether the trajectory analysis still uses the framework of Monaco 2 or not. And also, in the same time, they add clustering and also differential expression for the Monaco package. I have no idea why people do this. If you start from a comprehensive package, you should have an independent name and use Monaco as as one of the sub-function, but you name your whole package as a Monaco 3 and do everything. And also people use it as a trajectory analysis to, I don't know, that's their option. Okay, Slingshot is a relatively new package and start to show up and see in the comparison claims that you see the compared method have a Monaco, T-scan, waterfall, etc., etc. And you use this in different algorithm and for example, dimensionality reduction, different classes, supervised, unsupervised. The claims Slingshot is robust to noise. Well, again, this is just when they claim and publish a paper, and they can do this multiple linear inference. It look this path and also this path, and to be more comprehensively covery. So the three parts considered to recover the genes underlying. T-scan, I bring this up is the reason is the paper is well written, so you can look at the paper and re-implement everything easily. A stream, another package. So Lisa is from one of my collaborators. Anyway, so because we have so many and we need research, review, and side-by-side comparison between different algorithms. But what this paper does is group different algorithms based on their methodology, tree-based, graph-based. Bifurcation-based, bifurcation, linear method, circular-based, and different situation and to compare and use different parameters, okay? Accuracy, scalability, stable, usability, and have a comprehensive survey of all the methods available at that time. And then they gave this practical guidance for choose which method you want to use, whether your data construct, your cell population are relative homogeneous or more complex, whether you will consider artification tree or not. So you have different options at the end. Well, another R package has come out and they try to include all this available method to integrate them under the same package, so you can easily to use multiple algorithms to apply to the same same data set as a uniform input. So there are certainly the downstream analysis. Once you have the trajectory and what you can do here, I believe there's a review paper to do that. You can creatively use this trajectory. Here is one of the examples we are doing, trying to do here. So we have a population of cells and then align them using the trajectory here. And then along the trajectory, we can do a subsampling and do a sliding window to sample cells from the early stage and then move to another. Each window may have 500 cells. And then we can construct gene regulatory network for those. And once we have this multiple gene regulatory network, this create a data structure called tensor, and this we can use a lot of tensor related algorithm to decompose or denoise or learn from this structure. Okay, so this is one of the use of pseudotime to introduce some of these parameters, allow us to do this dynamic change of network. Another example of this design is, for example, you want to work on neurons, you can get from subpopulations and you can do a trajectory of pseudotime and then on the neurons and maybe also create a pseudotime theory, single cell gene regulatory network. Okay, so next we're going to do highly variable genes. So I will quickly show you how to get the highly variable genes. Okay. Single cell data, we have input matrix. So these are the cells. One, two. In this case, we want to focus on gene stuff. Disney or working on cells. Now, every data point is a gene. We want to characterize those genes. Genes expression variability. For example, if I have one gene, I can measure its gene expression across cells by using this R. So you have this. the higher means that cell expresses highly in that particular cell. Well, another gene have a different picture. It may have a higher somewhere, lower somewhere. You will get some picture like this. Okay. So in that case, we can compute the variance for this. Okay. And variance for this. It's clear that this variance is large, right? Variance 2 is large. Cross cells. The variability for gene 2 is higher than gene 1. Okay, if this variability is higher enough, we will see, okay, gene 2 is highly variable. Okay, is that clear? So there are many statistics we can use to measure the dispersion of variability. You can use variance, you can use standard deviation, and CV, which means standard deviation divided by it's called coefficient of variance. So since it's very easy for us, we just look at the variance of all the genes. We can do all the genes and compare its variance, and you put genes with large variance and tiny variability. Right? So the problem sounds very easy, and you can do that. First, we will see, okay, if you have a cell population have two different cell types. For example, we have half of B cell, half of T cell. In that case, you may expect some marker genes to show. So half of the expression of those of the B cells, the rest of T cells. In the B cell, you have a high expression of B cells. So in this case, any variable gene is a marker genes, more likely to be marker genes. Right? So you want to find a cell marker genes only express one type of cell type of others. So that automatically have some biological meaning for those tiny variable genes. That's right. So we want to use any variable gene to describe the sum. So here is a difference. We already done this. So the problem I'm going to consider is in this population, we don't have subpopulations. All cells are the same group. They all the same group. In my study, my own study, they all LCL in plus point cell lines. So, same cell. Now, our question is within the same cell, if you have highly variable gene, like highly, not highly, only variable gene, it's called a uniformly expressed. What that means, whether those genes are associated with any function will tell anything about this population of cells. So that's the topic we are going to discuss. In the middle, I will show you that we needed to have some control for those control for mean. In order to give more realistic estimation of the gene expression variance. Directly using variance have some problem with this lowly expressed, highly expressed. There's a lot of zeros here are cells which the expression level make it too long to be captured by single cell technology. Okay, in the middle, I will show you we need to have a correction of the way to measure gene expression variability for the gene of cells by controlling for average expression of that gene. Okay, is that clear? So as I said, we are considering a homogeneous population of cells, no subtypes. And those variability are between cells. So for particular gene, you measure expression in this cell and in that cell, very different. So we call it single cell difference in its gene expression. If you focus on particular gene, this will be single cell gene expression variability or single cell gene expression noise. In 2015, and this professor in Pennsylvania University, he proposed that the single cell, the difference, may be very important for its function. I mean, for the whole population of cells, the function. So this I call it the variation is function hypothesis. We want to test this variation is function hypothesis by looking at highly variable genes in a given cell population and see what those genes are, whether they will functionally reflect some of this cell's function. So because of the technology become available after we can collect data and look at variabilities across cells. Again, I have to emphasize the cells here are the same group of cells. There's no subpopulations. Our experiment get information from by the way, this is a computational experiment. So the lymphoblastoid cell lines, we sequence our cells. But the long airway epithelium cell, we collect from existing data and also dermal fibroblasts. And also, we have a new cell type. I'm not going to tell you right now, but later you will see what cell type we use in the study. Okay, to make sure we are measuring gene expression variability among homogeneous population of cells, we not only consider cell type, but also consider cells in the different cell cycle stage. So we can look at marker genes and see, okay, those cells are in the G1 phase, these are in S phase, and those are in G2 or M phase. If you look at the expression level overall, looks like S phase and G2 phase expression overall is higher. So because yellower means overall expression level is higher. That makes sense here because in S phase, the G2 phase cells are a little bit larger and also ready to be in the cell cycle, and compared to G1 phase cell relatively smaller, and overall expression maybe lower. So that's, you see the patch color tune difference. What we did is to get rid of all this phase of data. Only keep G1 phase cell. Eric, can you help me to get battery? Okay. Okay, so this is another round of cleanup of cells. Just want to make sure all the cells are in a similar or almost identical stage. No cell cycle effects, no cell type effects. We also filter out, discard mitochondrial expressed gene, and then focus on cells with a narrow range of library size. And also we did embedding, and then embedding and look at the manually pick a group of thousands of cells, which highly homogeneous. Okay, I don't have to explain to you what is embedding algorithm you use all the time, and I just want to show you the different algorithm applied to the same data set. You get a different visualization. So for lymphoblastoid cell line, there are cell lines, there's no structure among them. So you can confirm this by looking at Disney. No subclass, it's just hairball. Well, fate is more sensitive, and they try to push cells into different archistructure. They call it this branches. So to be conservative, we use fate to do this embedding, and then within the fate embedding, in the center, in the core of this structure, we just select 1,000 cells from here. Okay, so that's our selection of pure cell group. So that's a homogeneous population of cell you see here after multiple steps. And this picture is the same as what I draw on the blackboard here. And those genes have been ranked by their average expression. And these two genes have a very similar expression mean, but have very different expression patterns across cells. You see, among this nearly 1,500 cells, ribosome protein S11, this subunit, is a housekeeping gene, so it expressed in almost all the cells. No zeros, okay, cross. And but the expression level relatively low. I mean, it's just compared to this counterpart, but it's not so low at all. Yeah. Well, this hemoglobin gene, there are some cells has extremely high expression over 600, over 400 UMI. Well, a lot of cells has expression level nearly to zero or just zero. Yeah. No expression of all at all. But the average between these two genes are the same, almost the same, because one is ranked here and another is ranked a couple of gene down. Remember, we have several thousand of genes. This is just top 200 on the picture. So it's not a surprise if you compare the variation measured by coefficient of variance. Ribosome protein subunit have a CV equal to 0.43. Well, this immunoglobulin heavy chain the constant have a CV equal to 6.38, much higher. Okay. So if you plot the CV of these two genes, one is high, one is low on this scatter plot, which put CV and the mean at the same time. And these two genes have same mean, so they almost have the same X values, and also they all are log transformed. So these two genes have show this difference in their CV. One is high, one is low. Of course, this is a immunoglobulin, and this is the housekeeping gene, ribosome gene. So if you look at the relationship between CV and the mean, if we assume technical noise is Poisson distribution, and then CV this definition, and then if you do the log transformation, then log value of CV and the mu, the mean, should be linearly related. In our plot, we do have log CV because this is log scale and log mean. Okay. By definition, if the variability among the gene, among the cell for the gene follow Poisson distribution, and there's some highly expressed gene, so mu is high, you should have a lower CV. Okay, we expect to have a negative slope here, 0.5. So you expect to have this line. So this line is indeed this red line here. It's generated by expected technical variability follow Poisson distribution. So if you have a gene highly expressed, you have a CV lower, and if the measurement of this gene expression across cells only have technical noise, then all the genes should follow the right line here, regression. You can look at all the cells population, all the genes population. So each point here is a gene. You can actually model it better, use this nonlinear least square. So you have this green curve. Okay, so this green curve fit the overall data slightly better, more realistic. Another observation you can see from this figure is all the genes have the value distribution, which location of this gene always higher than this red line. It's always above this line. That indicates that for almost all the genes, the dispersion level is higher than expected by Poisson distribution. If it's purely Poisson, then those genes should be on this red line here. But if you above the red line, means your variability is higher than expected by only the Poisson distribution. Okay, so those variability are introduced by the biology itself. It's not technical noise. So we want to measure for every single gene here, the difference between its observed CV, was log transformed, deviate from it, expected from the technique Poisson distribution, which is the difference indicated by this yellow line, this from its observed to expected for this particular gene. So we can do this for all the genes, look at its different from the gene to expectation, which regressed either using the red or line or the green curve. So we actually use the green curve, because the green curve fit the overall values better, especially for those highly expressed genes at the right side here. So the difference is called residual Cv. So residual Cv is used as an indicator of the expression variability of each gene in the population. So that's why I think we need a control for the mean. So that's the way you control for the mean. You only use the residual of Cv, not use the Cv observed itself. And you can fit that curve using generalized linear model, and there are some procedures we can use as internally because the linker functions will allow you to model this curve. Okay, so we use generalized linear model. We use a correct linker function to model this distribution to fit this relationship. Now we can look at residual Cv for the genes. Here I select some examples. Some of the cells genes is above this curve, some are lower than this curve. So those genes are way above this curve are highly variable genes. Those ones are either housekeeping genes or more uniformly expressed across cell population. We can look at their profiles of the expression. So we have TMEM9B here, which is lower than the curve. We also have RPL, some protein large unit 17, and also below the curve. And its profiles like this, where the rest of the gene is way above the curve of expression like those. And you can have some impression of how the scale is once you look at the value of this. And here, for example, FTL, you have a higher, nearly over 1,000 UMI, while the majority of the genes is below 30 or 25 here only. So you have the scale, the feeling. That tells you that those highly variable genes, once they are expressed in those cells, the expression level is extremely high. But once they not expressed, it just shut down in the cells. Okay, so these are the highly variable genes. Any questions for this? Okay, that's the way you detect highly variable genes. So let's look at what genes are highly variable genes in lymphoblastoid cell lines. So those are the top 50 highly variable genes we detect after this filtering of cells and keep them consistent population, homogeneous. And those genes, if you search in ontology or in richer pathways or the immune system related response, cytokine medium signaling pathway, those genes are highly variable expressed in lymphoblastoid cell lines. And this cell lines itself is derived from the B cell, so it makes sense. They're functionally related to the immune systems. Well, remember we have highly variable gene identified from this long airway epithelium cell. And in those group of cells, these are the highly variable genes, top 50, and their functions. Some of still have development processes, a very large group of the function, but there are some characteristic pathways can be used to describe those genes, including this collagen formation. Remember, it's long, and you do need some of this production of collagen, maybe in the epithelium level. So to me, these are the reflection of the function of these cells. Let's look at thermal fibroblasts. Exactly the same analysis, repeat on the different population of cells. And here, the highly variable genes are those. They're different from other groups, okay, from other cell types. The most interesting and most important function to me is keratinization, okay, to create keratin and for the dermal, the skin, right? So this is also a unique feature of function for this group of cells, this genes reflected. So we can actually, from highly variable genes, to directly derive their function of cell types. So for epithelium cell, you need, in lung, you need a collagen formation. Well, in dermal fibroblasts, you want to have a keratinization. Well, in lymphoblast cell line, you have cytokine signaling, which has a representative function of those cell type. You don't want to mess them up. You don't want to have a keratinization in the lung epithelium cell. That was wrong. So in that case, I just show you in using this case study that highly variable gene itself contains very cell type specific function of that cell. So you probably should use highly variable gene to study cell population. well, what this cell type I just mentioned, and that's the min versus CV log transformed scatter plot. You can see from this particular cell type, this group of cells, the features of this is there's no highly variable gene can be detected in this population. So the picture looks very different from a LCL, from a dermal fiberglass, from a long epithelial cell. So all those cell types, you see some of this highly variable gene. Well, this one, all the genes follow this regression curve closely. There's no highly variable gene at all. Yeah, so these are the induced pluripotency potent stem cells, iPSCs. So for the differentiated cell types, they all have registered or committed function, and they have already had the iPSC just have no any functions you can identify because there's no highly variable genes. The other way around, that highly variable genes may be the most important genes which help particular cell types to perform the function of those differentiated cells supposed to do. Okay, highly variable genes are the most important ones. Those undifferentiated stem cell, there's no cell function or the cell type specific function have been assigned to stem cells yet. So there's no highly variable genes in there. So if our hypothesis is true, and we should be able to test using another cell population which is in the middle of the differentiation. So iPSC you can differentiate into neural stem cell. Neural stem cells in the middle of more differentiated cell types, astrocytes, oligodendrocytes, neurons, etc. So if we do this analysis using this cell type half differentiated, we should see some genes start to show this differentiation, become highly variable genes, right? So that's exactly what we see from this neural stem cell. You have some of the genes start to show high variability among cells here. Okay. Yeah, so we can also apply this to clotting sites, and this is a very important cell type, which is epithelium cells in the bile duct, and the highly variable genes are enriched with the function of this response to corticosteroids and glucosteroids, and also this different kind of a steroid hormone. And this is a very specific epithelium cell, and the function actually you can search the literature, and the most important function is for this clotting sites is to sense the stimulation from the hormones. Okay, so that's again is a positive example. So we can use highly variable genes to characterize highly specific function of cells. So the take-home message is single cell study, single cell data allow us to look at highly variable genes to measure the expression variability in the cell population. The cell population, I mean homogeneous population of particular cell type. This measurement, this readouts should be treated more seriously in the functional study of cell type. Because different genes have different levels of this variability, and the different cell types have different size of highly variable genes. And this function of the highly variable genes can tell you a lot of the cell type specific function. As I said from the beginning of the lecture, this message has been underestimated, underappreciated so far. And I strongly encourage you to try highly variable gene analysis when you have particular cells you really want to study its function. Okay. Questions? Okay, no? All right. So we will try highly variable gene detection Thursday using MATLAB tool. Okay. See you Thursday.